Okay, now as we just started talking about, the power to anti-age your brain, which is something I need to do on a regular basis. I feel just as I did when I was a teen, but I can feel aspects physically, but now at times I have what I call the Etch-a-Sketch moment. I'm about to say something. This isn't good when you're on radio, I've got to tell you. And suddenly, as you're about to say the word or the concept or the sentence, someone just goes, and what you're about to say disappears out of your brain and you're left with a blank mind. It's not a good thing. It's not a good place to be. Dr. Gawian McColl is head of the Aging Laboratory at Florey Institute of Neuroscience and Mental Health. Welcome to the program, Gawian.
Good afternoon. Thank you for the invitation.
It's very good to have you because you're going to talk about the effect of ageing on the brain and some of the latest research into how perhaps we can maybe hold that at bay. Maybe we can just have a little more, well, you put it best. What can we do to stop the ageing of our brain?
Well, I think speaking as a researcher, what we really need to do is understand the fundamental process of ageing. So what is it about ageing that impairs our function, impairs our ability to function as we could when we were younger? And to really conquer that, we need to understand ageing in something a little bit less complicated than a human. In fact, a whole lot less complicated.
Well, one of the things I was going to ask in the sense, and I'm sorry if I've interrupted you, but I find now, I mean, I used to do this a bit, but I'm finding now when I look for an example of something, particularly if I'm in a passionate moment where I think, yes, here is the answer, and it's as if that information isn't in the cupboard in my brain that I thought, and I go to say a name or a concept and it falls short, and sometimes I can scramble and find it, but it's not there on the tip of my tongue as I felt it used to be. Is that an aspect of an ageing brain?
I think it probably is. I think a lot of the information is still there, but our ability to connect to it and I guess make the connection and remember specifically does seem to become impaired. Yep.
And what are some of the factors? Obviously, is it things like the use of alcohol? Are there things that we are doing that are sort of preventing some of that information? But obviously, there's also the aspect of us it's a natural process. We're getting older.
It's the culmination of all of these things, really. Yep. So it's our genetics, which I guess is something that's more closely aligned to my research topic, as well as environmental factors. So, of course, human aging is quite an incredible thing. It takes decades to occur. And so we really have a long time for environmental influences to have an impact later on in life.
Now, an aspect of your research is the use of worms. Now, this isn't some kind of strange sort of you need to ingest them or anything. This is something where you're observing worms and Tell me about your research and how worms are an important part of this process of understanding ageing in the brain.
Yep, happy to do that. So the worm that my group uses is a worm that's used in many laboratories across the world. It's got an interesting name. It's called Caenorhabditis elegans, which is a mouthful, so we might just simplify to C. elegans. And this is not a segmented earthworm. This is a round worm, so it's microscopic. It's already found all across the world in the topsoil. It's an interesting fact. It's actually a reformed parasite. So at one stage in its evolutionary history, it was a parasite. It is no longer. It just lives in the topsoil, eating bacteria and making more worms. But the interesting thing about this animal is it has some traits about its biology that make it a really good, useful tool for looking at fundamental questions in biology. So it's been widely used in developmental biology, widely used in neuroscience, because it has 1,059 cells in total, and 302 of those are neurons. And so we understand an awful lot about how each of these neurons is connected to every other neuron, effectively making the brain of the animal. And so specifically for my research and for other groups interested in aging, one neat trick is the animal only takes 2 days to develop from an egg to an adult, 2 to 3 days. And then on average, it lives for about 2 weeks. So as an experimental system, it's a nice change from working on long-lived animals. This is a very short-lived animal. So if you're a researcher who likes to conduct many research within your working lifetime, something that lives only 2 weeks is a really good starting point.
So is this almost like a time-lapsing of how ageing occurs with our brains?
That's right, actually. So a lot of the characteristics of the way C. elegans age, maybe I should swap that around. A lot of the way humans age is mirrored in C. elegans as they age.
Right.
So they lose muscle structure, their neurons start to degenerate, they accumulate fat. So really they have a lot of similarities. They get dysfunction with their ability to move, they can't digest their food correctly. So all of these kind of degenerative processes are mirrored in worms, but it's in a truncated time frame. So we have time to study it, and we have time to intervene and try and do something about it.
And what are some of the things you've done to intervene?
Well, I guess I'm standing on the shoulder of giants here. So it was really back in... C. elegans has been used as a research tool for decades, but it was really in the mid-80s it was first identified that single gene mutations in this animal could double the worm's lifetime. So we could half the rate at which these animals age by changing a single DNA base pair in a single gene that made a single protein. And from that, we now understand that small genetic changes can have profound effects on animal lifespans. And so these pathways were identified in C. elegans, because now we know of many more genes that can do this, are very conserved pathways. So they encode the same kind of proteins doing the same kind of functions in humans. And so part of this is now our understanding of how insulin signaling works. This is intrinsically involved in coordinating our metabolic processes and things like our repair processes. And that kind of has a massive influence on lifespan and aging. And from that basis, I started to ask the questions, well, we can't really change our genetic makeup. So what are the fundamental cellular processes that are occurring during this aging event, and what can we do about it? And from that, we identified hyperaccumulation of iron as being potentially a very important factor.
So there's a build-up of iron that is not beneficial. Is that what you're saying?
That's right. And so it's really interesting. Iron is not a simple thing. So it's absolutely essential for all animals to have iron. And we have, speaking for all animals, we have a voracious appetite for iron when we're developing. The developing brain needs a huge amount of iron. But eventually our need for huge amounts of iron starts to plateau. However, animals haven't really evolved a sensible way of getting rid of excess iron. Once they're post-reproductive, once they're an adult, and once they're really beginning to age, we still have this voracious appetite. Our cells take up a lot of iron. And now the problem is that we don't store it correctly.
Right.
And so we have no systematic way of, or regulated way of getting rid of excess iron. So just like in C. elegans, humans and every other animal in which it's been looked for, there's this accumulation of iron with age, and eventually that starts to become a problem and starts to trigger certain damaging processes in the body.
So I guess the question is, what can we do to eliminate the iron?
So the kind of work that I'm specifically focused on is what does it do to the cells when they start to mismanage their iron stores? And what we think is it's triggering a particular kind of cell death process that's only recently been identified. And that process is called ferroptosis. So it's a way cells, normally with their iron load, if they sense they're becoming cancerous or if they sense they're becoming infected, they might trigger their cell death. It's a way of sort of stopping the spread of these particular conditions. And what happens in aging is this trigger for those cells to die might be switched on in error because of this accumulation of iron that we really don't need and we really don't want. And so what we can do about it, I think it's really exciting. I think this is a forefront. This is a point where we're looking to be able to do this in the future, is we might be able to think of ways of intervening and suppressing this problem, this trigger to die when you don't need to die, that's iron mediated.
What role does diet have within the accumulation of iron?
Well, it's critical, but our bodies are very, very good at taking up iron. So I think limiting your dietary intake is not really my research focus, and certainly being anemic or iron deficient is no solution to any kind of health outcome. But it's more tricky than that. It's really what iron you have has to be maintained correctly. So the body has a pretty good ability to buffer its iron reserves, but eventually that buffering seems to break down. And the iron starts to, in some ways, becomes a bit more mischievous. It can start interacting with things that it shouldn't normally be able to interact with, and that starts to cause sort of damage to the body.
And I guess there's obviously a very big difference too between someone who is slowly aging and their ability to pull that information out and think of things, because that seems like a natural slow decay, and then someone who has, for example, Alzheimer's. There's a very big difference between the two. Both can have an aging aspect of the brain. Is there a difference in iron levels in the case of someone with Alzheimer's?
Well, that's a really good question. So we have a fantastic team of researchers at the Florey, and my colleague and close collaborator Ashley Bush has had a lot of input into this thinking, and really his team, as well as another colleague, Scott Ayton at the Florey, first identified that the iron load in the brain is particularly elevated in Alzheimer's disease patients. And so we think this is a scenario where in Alzheimer's disease we don't really know what causes the disease, but I think we're beginning to understand what sort of controls the rate of the disease progression, and iron in the brain seems to be a really good candidate for this.
So what's the next step then? You sort of have identified these things, but what practical ways, moving from the roundworm as the I guess the architect of you discovering this to how this can apply to humans? What can we do next?
Well, I think we need to support research in this area primarily. This kind of research, although the animals only live for 2 weeks, it still requires careful work. So we need to be all behind our Australian researchers and behind the kind of world-class research that we're capable of. What we need to do is understand exactly what are the signals that these kind of changes in the brain are triggering. And then it becomes a process of what can we do about it. So what kind of drugs we can use to intervene and prevent these signals. So we're right at the point where we've identified that it's possible, but the kind of compounds and interventions that my lab is working on are really proof of concept. These are compounds, these are not drugs. We need to translate our primary observations into a translational pathway so that we can come up with compounds, or we can come up with drugs down in the future that can really target these things in a safe and efficacious manner. So I say we're not quite there yet, but we are getting there.
And the fact that they have a 2-week life cycle, it sounds like, as I said, it's a time-lapse, incredibly sped-up variant of the human brain. So is that difficult to monitor in that short time frame, all this information?
No, it's a sweet spot. So when we apply, I didn't actually mention this, when we apply the interventions that we're studying, we double the worm's lifespan. So they basically go from animals that live for, let's say, 14 days to animals that live about 30 plus days. So that's about reasonable for your average scientist concentration span. We can follow experiments for about 30 days. That's no problem.
And it sounds like the roundworms are a little happier getting a doubling of their life. Dr. Gawian McColl, thank you so much for your time today.